You may want to take advantage of radioactivity if you're studying biochemistry like me. So let me introduce you to the radioisotope. I hope I can convince you that they can be pretty dope. They behave like normal versions of atoms in almost every way, but their nuclei are uncomfy so they can undergo radioactive decay. In this process, the nuclei rearrange and change and let off waves of energy, and if we can't detect them, we can molecule see. So take that normal phosphorus, swap it out for P32, and now you can track what molecules do. You might think radiolabeling's a thing of the past, but I'm here to try to convince you with a technique that'll last.
We'll go over more of this in a minute, so don't worry if these terms don't quite make sense yet. But basically, molecules, so like everything we're interested in, DNA, protein, all these things, they're made up of atoms, which are these smaller parts. And so like individual carbons, hydrogens, oxygens, phosphorus. Phosphorus is, so that P, when I was talking about P32, that's a version of phosphorus that's radioactive. So like atoms can have different versions of themselves called nuclear isotopes. And some of these are radioactive, and so that means that they can undergo that process where they let out that radiation that we can detect. So if we swap out radioactive versions of these atoms for the normal versions, we can label molecules, and this allows us to track them. So it's really useful for doing things like labeling the ends of an RNA or DNA to track them, or like labeling like tracking kinase activity. So kinase are these molecules that add this like phosphate group, so like this phosphorus surrounded by oxygens to other molecules, and this like changes their activity and that sort of thing. So if we use a radioactive version of phosphorus in that, we can then track the kinase activity. And so this post is actually like a prequel to a post I'm going to do telling you more about the kinase assays. But today I just want to cover the background of radioactivity and this radiolabeling and stuff. And so you might wonder, like, isn't radiolabeling like a thing that they did in the really old days before they had things like fluorescence? So fluorescence is basically where a molecule will give off light. And so when you're just giving off light, it's not as potentially dangerous as radiation. But it's a lot less sensitive, and it adds this like bulky group to a molecule, whereas with radio labeling, you're basically just changing an atom that was already there. So to all the other surrounding molecules, it looks exactly the same, but to us, it is detectable in its like native state. And it's super, super sensitive, way more sensitive than fluorescence, or any like dye or that sort of thing. It's really, really sensitive. And in terms of the safety, we take a lot of precautions with the safety, but the amounts of radioactivity that we're working with in the lab are really small, really, really small. And it's a type of radiation that you can block easily with like some thick plastics. We have like plexiglass dividers and that sort of thing. We also have little monitors to make sure that we don't our doses don't get too high. So we have like a little ring we put on, and then like a little badge that we can put on for like whole body radiation. And so then like every month the safety people come and they collect them and give us a new one, and then they make sure that we're not being exposed to too much radiation. But yeah, there's radiation everywhere in the atmosphere, and we'll talk more about this. And so the type of radiation that this is tracking is like ionizing radiation. And so ionizing radiation is like super strong energy, so it can actually like break electrons off of molecules, and so that's really dangerous when it's doing things like breaking DNA and that sort of thing. But in terms of how we can detect it, so basically electricity is like the flowing of electrons. So if you can break electrons off of other molecules and get them to flow, you can get them to flow like between two detectors. So like for a Geiger counter, it has like this tube, and then it has this shielding around it, and inside is like an inert gas, so it's like typically non-reactive. And then if you have ionizing radiation, it comes and it breaks off electrons, and then those electrons can travel between the rod and the outside thingy and let off an electric signal, and that's when you get like a beep or a crinkly noise with the Geiger counter. And so we have like scanning Geiger counters we can measure. We also have, I've showed you before, like we have those screens where we can measure the radioactivity that's let off in like the storage form, and then scan it with the laser later and let off light. And we also have this thing called scintillation counting, where we put things in little vials in this in this machine that I'll tell you more about in the kinase activity post. But today let's talk more about radioactivity and what all those like jumble of words I meant. Molecules are made up of elements, which are things like hydrogen, helium, carbon, nitrogen, oxygen, phosphorus, sulfur, all these different things. The main ones we deal with in biochemistry are like hydrogen, carbon, nitrogen, oxygen, phosphorus, sulfur. These are like our big guys. But there's all these different elements. And what makes each element unique is its number of protons. So elements are defined by the number of protons. So what are protons? Okay, let me step back a second. And so atoms are these really, really tiny things, but they're made up of even tinier things, subatomic particles. And there's three main ones. There's the protons, which are positively charged, neutrons, which are neutral, and electrons, which are negatively charged. And so the protons and the neutrons hang out together in this dense central core called a nucleus, and then they're surrounded by this giant cloud of these negatively charged electrons. And these electrons are really tiny, but they're really important because basically everything in chemistry and biochemistry comes down to the idea that opposite charges attract and like charges repel. And so even when a molecule doesn't seem like it's charged, because it has an even number of protons and electrons and they have equal and opposite charges, so those will cancel out, you get a neutral molecule, but you can still have partial charge based attractions because those electrons are in this like cloud and they can spend more time in various regions, and they can also interact with electrons from other molecules, especially like the outermost electrons, which we call the valence electrons. So they're farthest from the protons, and so the protons are this positive force that's kind of like pulling in, reining in those electrons, but the ones on the edge, so those like valence electrons, are a little promiscuous, and instead they can interact. And so when you have atoms like interacting, and if they like share their electron clouds, they share like pairs of electrons, that's how you can get these like strong covalent bonds, which is what holds molecules together. And then the molecules, they can still have this like regions of charge and that sort of thing, so they could be attracted or non-attracted. So these electrons are basically, we focus mostly on the electrons in biochemistry, and this radioactivity is kind of like where the nucleus is like, hey, don't forget about me, I'm pretty cool too. So speaking of that nucleus, we have that number of protons, and so the number of protons defines the element. So for example, phosphorus has and it's called the atomic number. And so phosphorus has an atomic number of 15, which means it has 15 protons. Carbon 6, it has 6 protons. Oxygen is 8, it has 8. And so each of these elements, and that's how the elements are aligned in this table, the periodic table. So the atomic number is that number of protons, and that's what defines the element. So if you have 8 protons, it's going to be oxygen. If you have 7 protons, it's going to be nitrogen. If it has different number of protons, then it's not nitrogen. But the number of electrons and the number of neutrons can change. So if we change the number of electrons, we change the charge of the atom. And so if you have an imbalance of protons and electrons, then you get a charged molecule, which we call an ion. And so you can have, if you have more electrons than protons, you get an anion, which is negatively charged. The other way, if you have more protons than electrons, you have a cation, which is positively charged. So, and changing the number of electrons, it doesn't change the identity of the atom because it still has the same number of protons. But it does change the properties of the atom and how it interacts with other atoms because, as I said, this charge is like super, super duper important. So we talk a lot about like ions and charge and electrons and stuff in like past posts and talks, and that we focus a lot on that in biochemistry. And the protons we also think about because that determines the elements. But you can think of every little click of the Geiger counter as the neutrons calling out, hey, don't forget about me, I'm important too. And so radioactive decay is one place where the neutrons are going to finally play their starring role. And so we don't really think about it that much in biochemistry, and so I'm not going to try to get into like the physicsy details of what's going on. But the basic idea is that you can have atoms can have different numbers of neutrons, and we call those nuclear isotopes. And some of those are radioactive, so some of them are unstable and they will let off this radioactive decay, and some of them are just heavier or lighter. But the reason why, or like one of the reasons why you have neutrons, you might be wondering, like, what the heck is the point of having these neutral things hanging out in an atom? Well, it turns out that they were actually really important. They serve as a sort of glue for holding the protons together. So if we go back to the idea that like charges repel each other, and the protons are all these positively charged things hanging out together, you're going to need some sort of glue to keep them from like breaking apart. And that's where the neutrons come in. When you have lighter elements, so things like carbon, nitrogen, oxygen, the like quote unquote normal, so like the average version you'll find is like it normally has like the same number of protons as neutrons, or around there, because you don't need that many neutrons to rein in the protons. When you get up into the heavier atoms, however, you have a lot more protons that you need to glue together. And so you might need more neutrons. And so when you have heavier atoms, they often have like a lot more neutrons than they have protons, because the neutrons is acting as a glue through this thing called the strong nuclear force that I am not even going to try to explain because I have no clue really. And I think there's a lot that's still unknown about it. But basically, you can think of the neutrons as serving as this glue that's holding the protons together and keeping the nucleus from like splitting apart, because all those protons want to get away from one another. But sometimes the neutrons lose, and sometimes the nucleus isn't stable and it can let out radiation. So radiation is just like these like waves of energy, and so unlike the radiation that's like light, like this is way like the visible light that we think of, like that's a form of radiation. It's energy waves, but this type of energy wave is way more energetic, and there can actually be like particles and not just radiation. So there's a few main types of radioactive decay. So there's like alpha decay, where it actually lets off this like alpha particle, which is like a helium nucleus, so it's like 2 protons and 2 neutrons. Beta minus decay, which is the main form we deal with in biochemistry, which is what like P32 does. So it takes a neutron and it swaps it out for an electron. So it takes a neutron and it like transforms it or transmutates into an electron, and then this weird like other thing called the antineutrino. There's beta plus decay, also known as positron decay, where it takes a proton and it turns it into like this positron and a neutrino. And so a positron is like a positively charged electron. So it's like super tiny, but it has a positive charge. So it's not influencing the mass, but it is influencing like does have a positive charge. And then you have gamma decay, which is it just like the nucleus kind of like rearranges itself, like it shifts around a little and lets off energy. So it like gets more comfy, and then it like, so when it's comfier, it's like in a lower energy, and so it lets off that energy in the form of this like gamma ray, which is kind of like an X-ray on steroids. So it's like super, super energetic. So this like alpha and beta decay, there's this like particle decay, because there's actually like particles in these waves of energy. And then gamma ray is just, well, I say just, but it's like this super energetic energy, but there's not actually like particles. But it's like this decay is called like ionizing radiation, because it's so high energy that it can actually strip an electron off of another atom, like out of the electron cloud though, so not out of like the nucleus. So it's kind of weird when we're talking about beta decay, because you're actually like generating this electron out of a neutron. But normally when we talk about electrons in biochemistry, we're talking about those electrons in this electron cloud. And so it's really confusing in nuclear chemistry, is that often the equations and stuff will kind of like ignore the electrons that we normally think about. So you're wondering like, where the heck are like the charges and everything? And because they're focusing on mostly on the nucleus, but the like charged particles that come out and stuff can like quickly join up with other like charged particles and grab back on electrons if they lost electrons and that sort of thing. So they don't really focus on them. And so you just like focus on the nucleus when you're looking at nuclear equations. That we use in biochemistry are like P32, so radioactive phosphorus, and like like S35, so radioactive sulfur. And you can see that, so they're letting off this electron and this antineutrino, but they're doing that by converting a neutron into that. Protons and neutrons are called nucleons, and so the number of nucleons is going to be your mass number, and then the number of protons is your atomic number. And so you see, like in nuclear chemistry reactions, the things are written like this. So like the normal version of carbon is carbon 12, and so it has 6 protons and 6 neutrons, so it would have a 6 here and a 12 here. And so often we just put, like, when we're talking about P32 for say, we don't really think about the 15 because the P kind of tells us like it already is 15, so we don't need to put that, but when you're doing equations and stuff, you want to have it there so that you can see what's going on. But you can see that because you're changing a neutron into a proton, now you're actually changing this atomic number, now you're changing the element. But the thing is, when we're doing like radiolabeling and that sort of thing, we only have like a tiny portion of the molecules actually radiolabeled, and there's only like so radioactive elements have a thing called the half-life, so which tells you like how long it will take for half of it to undergo decay. And so for P32, this is like 14.29 days. So like every couple of weeks, there'll be half as much. But that can still be a lot. And it's really sensitive. And since it's only a small part of what you're looking at, we don't really have to worry about this small portion of the molecules undergoing this decay, and then therefore like changing. But we're just worried about like, we care that we get to see it. But so what's happening is you can see that with beta minus decay, you have the atomic number, or you have the atomic number changing, but this mass number changing staying the same, because you're taking a nucleus and neutron and you're turning it into a proton, and so those both have mass, the same mass, and then you're letting off this electron. And then so similarly with like beta plus decay, where you have a positron being emitted, now you're going like back to P50, to silicon instead of phosphorus. And if you had alpha decay, then you're actually letting off like this whole like 2 protons and 2 neutrons and stuff. With gamma, you're not changing the identity at all because you're just letting off the energy. But I'm not really going to talk about nuclear equations too much. I just wanted to introduce you to that idea of like the mass number. And so just a quick thing, so this is like the mass number. It's different from the atomic mass. So like if you were to look up carbon, so carbon has a mass number, so like carbon 12 is a mass number of 12. But if you were to look up carbon on the periodic table, you would see it would be a little higher than 12, and that's because there's like some carbon 13 in the like in the environment, just like normally. And so that a version of carbon that has 7 neutrons, and it's not radioactive though. But there's also like carbon 14, which is slightly radioactive, but it has a really long half-life, so it takes a really long time to decay. And so the half-life is the time that it takes for like half of the molecules to decay, but it's like this random process, so you never really know which one's going to like decay or whatever. But that allows us to do things like carbon dating, where we can use like measure how much decay happened and then learn about like how long something had been dead or that sort of thing. But anyway, so that's so that would be like the average atomic mass that you see on a periodic table, but we're dealing here with mass number, which is like the number of nucleons, so protons and neutrons. We can take advantage of this decay, and so by swapping out a normal phosphorus on like the end of our DNA or that sort of thing, that we're measuring in the lab, and so we can make like we call this like end labeling. So we replace one of these phosphates, so like RNA and DNA, they have these like phosphate groups on their end, and so we can replace that phosphate with a radioactive one. So we can do this using like 5' end labeling. So we can, if there's a phosphate there, you have to like remove it first, or if it doesn't, then you can just go straight ahead. And so we use like a kinase instead of a phosphate adder, and it'll take this gamma phosphate, so this end phosphate, and it will add it to that. And now we have a radio label thing. We can use it as like a probe or that sort of thing. You can also like body label or whatever, so you can like change all the phosphoruses. So if you like make the RNA or DNA in, like, give it the phosphorus thing, like when it's growing, when it's being made. And so there's also different ways that you can label different parts of molecules. So when we're looking at, so with like proteins, you can change, like use radioactive sulfur. With nucleic acids, radioactive phosphorus. And that allows you to like distinguish between different molecules. And we saw this come into play when we were looking at the Hershey-Chase experiments, where they labeled the DNA, or they labeled the proteins, and then that allowed them to track which was providing the genetic information. So more on that in another post, and I'm not going to go into it too much. We've also seen like normal, like non-radioactive isotopes come into play when we've had hydrogen, or we had like nitrogen, heavy nitrogen labeling of DNA in the Messelson-Stahl experiments. And so they labeled some of the DNA with, they made it heavier, and then they could separate it by their heaviness. And so this brings us to the important point, going back to the idea of the nuclear isotopes, is that not all nuclear isotopes are radioactive. And so sometimes you can have like an imbalance, or like an awkward imbalance of neutrons and protons, which actually isn't that awkward, and they're happy with it. So like this nitrogen 15 has 8 neutrons and 7 protons, unlike like quote unquote normal light nitrogen, which is 14, so it has 7 and 7. But this is like heavy but not hot. So hot is kind of like a slang word we use for radioactivity. And so heavy nitrogen allows us to track molecules, but it's not radioactive. Whereas like P32, so the phosphorus, and the like S35, so sulfur, those are both radioactive. So they let off these this high energy radiation that we can then detect. And so yeah, so the great thing about radioactive labeling is that it's not actually like changing the molecule itself, so you don't have to worry, like it reacts normally. So it allows you to study the molecules in like a more native format. So especially for like when we're doing DNA or RNA, we don't want to have, we often like if you care about the end of the RNA, so like I work with a protein that binds to it binds to like the end of like the 5' phosphate group, and so I want to make sure that it's not like it doesn't have a fluorescence group there, because that would be blocking things, and that would be a problem. And so I buy gamma ATP. So ATP, so we label these, has 3 phosphate groups, and so they're labeled alpha, beta, gamma, in order, just like the Greek lettering for like the numbers, like how far you are from the actual like ATP part. And then the end one is the gamma phosphate, and so it's a version that's radioactive. And so it comes in this little like vial like this, and it has this like green stuff in it, so you can like see it better. But of course, what you really want to track it with is the Geiger counter. So we have like the scanning Geiger counter that's like this, and then we keep it on when we're dealing with radioactivity, and then we survey before and after. So we like scan that pancake thing over the surfaces and make sure that nothing's giving off too much radiation. There's always going to be some like noise, because there's ionizing radiation all around us from all sorts of different things, but we don't want to have like above background, basically. And so hopefully that helped convince you that radiation is still used and still useful.